Hello, I am Priscila Ramos Ibeas. I am a doctor in veterinary sciences, and I work as a researcher on embryo development at INIA, which is a national research institute. Well, early embryo mortality is a very big problem for both humans and farm animals. For example, in humans, it is estimated that around 30% of the embryos that are naturally fertilized in natural conditions would result in a live birth, with around 10% would be clinical miscarriages, which are the ones that we notice, and around 60% are embryos that would be lost before or around implantation, and this is a very high percentage. And these losses remain unnoticed because they happen before the missed menstrual period. So around this period of development, there are very important things going on in the embryo. For example, the cells are deciding which cell type they will become or which organ they will form in the future. And this depends on the expression of specific genes. So what we are investigating are the genes and signaling pathways that are operating during this moment of development. And we are using animal models like the sheep and the cow. So investigating the molecular mechanisms that are involved in early embryo development using animal models, it's essential to understand why the embryos are dying or why these early pregnancy losses. And then in this way, we could design strategies to avoid the pregnancy losses. For example, once we identify which are the important genes for fertilization or for embryo development, we could identify mutations that could be causing infertility in humans. And right now, or in the future, we could use, for example, CRISPR-Cas9 technology to try to correct these mutations through gene therapy. So I would say that we have two main innovative tools in our routine research. One is CRISPR-Cas9 technology that is a molecular tool that allows us to do precise genetic modifications from the thousands or millions of base pairs that the genome have. We can modify or cut in a specific one. And in this way, we can analyze the role of a specific gene. So in our routine work, we collect sheep and cow ovaries from slaughterhouses because they are normally discarded. They are not used for human consumptions. We extract the oocytes, we microinject them with the CRISPR-Cas9 reagents, and we fertilize them in vitro. Then we culture the embryos and we see what happens in the embryo when we remove a specific gene. And the second innovative tool that we have is a post-blastocyst in vitro culture system. So conventional embryo culture system only allow to maintain embryo development up to the blastocyst stage. We have developed a system for cow and sheep embryos that allows us to culture the embryos in vitro for 1 more week. So in this way, we don't have to use experimental animals to see what happens after the blastocyst stage. Well, there is another tool that I haven't mentioned that we are also using, which is called single-cell RNA sequencing. So with this technology, we can analyze thousands of cells that are present in the embryo, and we can know all the genes that every cell is using at each time of development. So we are generating like an atlas of embryo development. This has been done in mice, in humans, and we are also doing it in the sheep. And from this point, we select the genes that we think they are important to study their function. We identified one protein that we think it's important in the oocyte for fertilization. Then we generated with CRISPR-Cas9 a mutation to delete this gene. So when we remove this protein, we think that the sperm won't be able to penetrate the oocyte. So as we want to study the gametes, we need to produce the animals because in our routine work, we only study embryos in vitro. But for this study, we had to generate the genetically modified animals. And now the animal is born. We need to produce more, but we still have to wait until Teodoro is older and can have offspring. So the study is still in progress. Well, the dogmas in developmental biology or in embryology have been established using the mouse in the labs. So until very recently, until CRISPR-Cas9 arrived, genome modification could only be done very efficiently in the mouse. But in the last years, we and other researchers have investigating, and we are discovering that the mechanisms during early embryo development are very different between the mouse and other mammals. So we are studying embryo development during the first 2 weeks in other species like the cow or the sheep, because they develop in a very similar way to the human, which is a flat embryonic disc, and this is very different to the mouse. So I think these farm animals are very good models for embryology. And another advantage is that we can produce thousands of embryos from ovaries recovered in the slaughterhouse. And if we want, we can analyze the development until later stages than humans, because human embryos can only be developed up to 14 days in vitro. Yes, in fact, there are already some therapies, gene therapy, applied to some diseases in humans. But I hope that in the future they become more available to also infertility, for example. In theory, using base editing, which is an alternative CRISPR-Cas9 system, allows you not to cut the DNA, but to correct mutations. So if you microinject this in an embryo which has a mutation, it should be able to correct it. Also in patients, if the problem is in the oocytes or in the sperm, with gene therapy, you should be able to correct these mutations. You have to be very sure that you are only generating these modifications in the genome and not others, because sometimes there are regions in the genome that are very similar. So until this could be used, we still need a lot of research, and it also needs to be regulated very well before it can be applied. Well, achievements, well, we are doing so many genes right now. We are removing so many candidate genes. We have produced these first genetically modified lambs for the first time in Spain. But it's also very challenging work, because it's a very complicated process. Generating genetically modified embryos have many steps there are many steps that can fail. And also working in other species is more complicated because, for example, the reagents that we need for research, sometimes they are produced to be used only in humans or only in mice, and they don't work in other species. So, but as a lot of things in research is trial and error. Well, there are still so much to do. So in general, I want to know more about embryo development because, for example, as I mentioned, in the mouse, if we remove one gene, for example, the blastocyst doesn't form. In other species, we do the same and nothing happens. So we still don't know at the molecular level how a human blastocyst or a sheep blastocyst is formed. So this knowledge is essential for them understand why the embryos don't develop. But these mechanisms are so complex that I could be working on this for a lifetime, and still there would be so much to discover.